Welcome to my cooking show with on today's menu, the EcoTox Pasta Salad Delight. We'll start off with a base of copper noodles and prepare some cadmium carbonara sauce. Then we'll finish it off with some zinc-coated croutons and some bisphenol H cheese. Smells delicious, but what if I actually ate this salad? Pollutants that are frequently found in the products like I just showed you can often end up in the sea through sewage effluents or the atmosphere. Not only do they affect aquatic organisms, but they can be a threat to us as well. You and I probably like to eat seafood, but 3 billion people worldwide depend on seafood as their main source of protein. So it is very important to create ways to assess the effects of pollutants on the health of organisms. One way to do this is by the use of bioindicators, which are organisms that can be used to screen and reveal the health status of an ecosystem. My name is Frida Cnossen, and as part of the ERA team and under the supervision of Eric Ruttinger and Joao Carvalho, we are aiming to assess the effects of pollutants and pharmacological inhibitors on the regenerative capacity of Nematostella vectensis. This immersion project is sponsored by INSERM and takes place in the IRCAN, the Institution for Research on Cancer and Aging in Nice. Why the IRCAN? Because the effects of marine pollutants can eventually have an impact on humans, leading to diseases such as cancer. Nematostella vectensis, also called the starlit sea anemone, is an anemone living in the estuarine environment. It can tolerate high amounts of stress, and it has been used as a model organism to study its regeneration, as it can actually undergo full body regeneration after being injured. The pollutants that we are currently studying include 4 heavy metals, zinc, mercury, copper and cadmium, and 2 endocrine disruptors, bisphenol A, which is often found in plastics, and retinoic acid, a derivative of vitamin A that can be found in sewage effluents and microalgal blooms. So our goal is to establish Nematostella vectensis as a bioindicator that can visually respond to these pollutants by giving off a fluorescent signal. Now I'll give you a brief overview on how we do this. Previous students in this project have determined the LC50, the lethal concentration 50, for each of the pollutants, meaning at which concentration of the pollutant 50% of the Nematostella survived. In the next phase, several genetic biomarkers have been selected from the literature, and we are using the LC50 and quantitative PCR to determine which genetic biomarker has the highest response to a pollutant. During this project, we have also been working on developing a system called CRELOX-P that would fluoresce in different colors based on the presence of a recombination enzyme called CRE, and we have micro-injected this system into embryos of Nematostella to test if it works. On the left side you can see the green expression in the absence of CRE, and on the right side you can see the multiple colors when CRE was added. In the end, we could link this system to the pollutants by creating transgenic lines, so that Nematostella would fluoresce in different colors based on the presence or absence of a pollutant in the water. It is now up to future students to develop this system more and link the various pollutants to the CRELOX-P fluorescent system to establish Nematostella as a successful bioindicator.